Welcome back to MLT 240. In this lecture we'll be covering cestodes, which is also known as our flatworms or our tapeworms. This lecture will be divided into two parts. So we just need to first go over some general information for cestodes. The phylum is Platyhelminth. They are long, ribbon-like, flattened worms. If you were to cut them in half, like we talked about cutting the roundworms in half, the cross sections of flatworms would be flattened cross sections. The internal organs are bedded in tissue called parenchyma. The parenchyma for flatworms cause the flatworms to really not have a cavity for internal organs, so they don't have a respiratory or blood vascular system. And cestodes are hermaphroditic, meaning they have male and female reproductive organs within the same organism. Adult cestodes live in the intestinal tract of vertebrates, which are their definitive host. The larval stage inhabits tissue of the intermediate host. The oncosphere, which is a tapeworm embryo, can be seen within the tapeworm eggs, and it has 6 hooklets that facilitate entry and attachment of the embryo into the intestinal mucosa of the intermediate host. The oncosphere can also be referred to as the hexacanth. Hex meaning 6, referring to those 6 hooks or hooklets that are there and available for attachment. And a little bit later on in the PowerPoint, you'll be able to see an image to see exactly what those hooklets look like. Now let's just go over some general anatomy for our cestodes or our flatworms. First, let's talk about what the scolex is. It's the anterior end of the adult flatworm. It attaches to the wall of the intestine of the host by means of suckers or hooks. And we'll see what that looks like here in just a minute in an image. The proglottid is one of the segments of a tapeworm. The proglottid contains the male and female reproductive organs when mature. A gravid proglottid is the reproductive organs in which the proglottid matures gradually, so that proglottids toward the terminus end of the tapeworm contain fully developed reproductive organs, and the uterus is filled with fertilized eggs. The shape of the gravid uterus is distinctive of each species and can be found singly in just one or in short segments broken off in feces. So the gravid proglottid sections is usually what you're going to find in your feces or stool samples. The tegument is the body surface of Platyhelminths. The cestode's tegument is the site of nutrient and oxygen absorption, as well as the site of waste excretion. The strobila refers to the entire body of the tapeworm. The rostellum is the anterior protuberance of the scolex of some species that may bear a circular row of hooks, or it may be retractable, and we'll see exactly what that looks like in some images coming up. So let's look at some images of a sexually mature proglottid and a gravid proglottid. Remember, our tapeworms are hermaphroditic, meaning they can have both male and female organs. So if you look at this sexually mature proglottid, you can see a uterus, you can see testes, you can see ovaries, so you have both male and female organs on the sexually mature proglottid. Now our gravid proglottid, meaning that the proglottid is not only sexually mature, but it has fertilized eggs. Here are some fertilized eggs on the sexually mature proglottid, but we can also differentiate between the different species of tapeworms based on the pattern of the gravid proglottid or the uterine branches, and the eggs are within the branches of the uterus. So on this slide, let's look at the difference between our immature proglottids versus our mature proglottids. And remember, for proglottids, we're talking about the different segments of a tapeworm. So the proglottid is a segment of the tapeworm. So here is the scolex, or the head of our tapeworm, and just imagine this tapeworm being one whole tapeworm, and it's kind of folded up like this. We're just going to imagine this is a continuous tapeworm. So we're starting all the way down here at its head. And as it goes along, it's going to mature more and more. Okay? But right here, these sections or proglottids are smaller, so they're still immature. But as you go along, you see how the proglottids or sections are getting a little bit larger. So these are what's considered mature. So the sexual organs for both female and male organs, because again we're hermaphroditic, these are mature at this point. But if you continue on, once they're mature, we can start producing fertilized eggs. So these are the fully mature proglottids or sections that are full of eggs. So we're out at the very end of the posterior end of the tapeworm. So these sections are going to be able to start falling off. And these little sections or proglottids are what you're going to be able to see in the species. And if you look real close here, you can see the different uterine branches that are branching off that are holding all those fertilized eggs for our tapeworms. So let's point out a couple more things for anatomy. Again, we were talking about the different sections of the tapeworm. Here would be the head, and as you go along the length of the tapeworm, again, you're going to mature, and the ones on the end are going to be completely mature with fertilized eggs, and those sections are going to be ready to fall off into the fecal samples or stool samples. But if you zoom in on the head, okay, we're going to look over here and we're going to name some of these different parts. So the head as a whole, is referred to as a scolex. Okay. Within the head or on the head, some tapeworms have suckers that help them hold on, and some have these little hooks, and those hooks are on the rostellum. Okay. If they have any hooks, they're located on the rostellum. Also, there's the neck, and of course, the immature proglottids would be the closest up to the anterior end of the head. Now let's talk about some general pathology of our cestodes. This first little section are all larval stages of infection. So infection occurs by ingestion of larval stages. First, there's the cystocercoid. It's a thin-walled, fluid-filled bladder-like cyst that encloses a scolex. Then there's the cystocercoid. This is a small bladder-like structure containing little or no fluid in which the scolex is enclosed. Then there's the procercoid. This is the larval stage that develops from the choriocydium, which is very much like the oncosphere or the embryo of D. latum. It develops in the body of a freshwater crustacean. Plerocercoid is the larval stage that develops after a freshwater egg ingests the procercoid stage, has an immature scolex, is infective if eaten by a human. After an embryo hatches in the intestine of the intermediate host, it can migrate through the intestinal walls to a specific tissue site and can be found in undercooked meat and fish or in intestines harboring the larva. In our first two cestode species, Hymenolepis nana and Hymenolepis diminuta. These are known as the dwarf tapeworm and the rat tapeworm, respectively. They're transmitted by ingestion of eggs in contaminated food and water, or they're also caused by auto-reinfection by hand to mouth. Light infections of both its species are often asymptomatic. Heavy worm burden of H. nana can cause intestinal enteritis or inflammation, weakness, headaches, anorexia, abdominal pain, dizziness, and diarrhea. Young children a lot of times get misdiagnosed of having pinworms or anaerobius vermicularis because of the symptom of having an itchy bottom. Here is the life cycle of Hymenolepis nana. Humans usually become infected when they ingest that larval stage, the cystocercoid, of an infected arthropod. For H. nana, the adults will live in the small intestine H. nana can cause a retroinfection with no intermediate host required. Diagnosis of H. nana usually happens through eggs being in feces or found in feces. For the species H. diminuta, eggs are usually found in the feces of infected mice or rats, and that's the common source for human infection. Humans accidentally ingest the contaminated mice or rat fecal matter. Also, another way humans are accidentally infected with H. diminuta is they accidentally ingest fleas or beetles that have the H. diminuta larva stage. So let's talk about what H. nana would look like under the microscope. You would have a colorless shell, so you shouldn't see any color. There would be filaments emerge from polar thickening. So this little stringy material would be your filament. You would have hooklets. These lines right here would be your hooklets. We're going to be able to see hooklets a little bit better in the next image. And this is known as a hexacanth embryo because of the 6 hooklets. The size would be 50 to 30 microns. The H. diminuta, the eggs would be round to slightly oval. They're a little bit bigger. They're anywhere from 70 to 86 microns to 60 to 80 microns with a striated outer membrane and a thin inner membrane. So here is our outer membrane. Striated meaning it kind of looks like it has stripes or striations, and a thin inner membrane, and there's space between membranes that is smooth or faintly granular. So in this area it could look like there's grains or granules, and the oncosphere has 6 hooklets. So the inside of the embryo would have 6 hooklets. So there's 1, 2, 3, 4, 5, 6. So those are your hooks. Here's some examples of Hymenopus as far as worms or mature worms. Here are 3 examples. Here's the heads. The little small end is the head or the scolex. And of course, as you travel along, those proglottids are getting more mature, so they're able to fall off. That's part of how they're going to reproduce or keep producing more worms. So as you go along, you're going to get more mature for the proglottids. So the head end is going to be smaller, and as the sections go farther out towards the posterior end, they're going to get bigger because they're more mature. Again, here's the head or the scolex out towards the posterior end. They're going to get bigger because they're becoming more mature and getting ready to fall off. Hymenopus is the most prevalent of all tapeworms in the U.S. It's encountered worldwide. It's common in the southeastern U.S., the tropics and the subtropics. In temperate areas, its incidence is higher in children and also amongst institutionalized groups, people that are in close quarters or people that are living in areas of poor hygiene. It requires no immediate host. A common definitive host is a mouse. H. diminuta, while less frequent, has been reported from various areas of the world. Now let's move on to our next species of cestode or tapeworm. It's Tinea signata. This is known as our beef tapeworm. The transmission is eggs are eaten by the intermediate host or a cow. Humans ingest the undercooked beef from the infected cow. That's how we become infected. Light infections of T. signata may be asymptomatic, but heavier infections may cause abdominal pain, diarrhea, weight loss, and moderate eosinophilia. Occasional appendicitis or cholangitis can result from migrating proglottids. Some 60% of patients can end up having cysticercosis or having larvae in the brain. Signata is usually found in the southwestern U.S. or in countries where beef is eaten raw or undercooked. This is the life cycle for T. signata. You get it from eating the undercooked beef, but this particular diagram is showing the life cycle for T. signata and T. solium. We'll be covering T. solium here in just a minute, but the cyst is embedded within the meat, and when it's undercooked, that's how the humans become infected. As far as lab ID for Tinea signata, the diagnosis, we usually see the eggs or proglottids in feces, but after treatment, you might see the scolex or the head in feces, where the head has released from the intestinal wall and it's passed in the stool sample. In T. signata, there's somewhere between 15 to 30 lateral uterine branches in each side of the gravid pyglottid. The scolex has 4 suckers, so the head, the scolex has 4 suckers on it, and the rostellum is known as unarmed, so it doesn't have any hooks, and we'll see what that looks like here in just a minute. The eggs of T. signata, the beef tapeworm, and T. solium, the pork tapeworm, are identical, so you're really not going to be able to tell the eggs apart under the microscope. You're just going to be able to say, yes, this is a tapeworm egg, but I'm not going to be able to identify the species based on the egg. We are able to do immunologic testing methods for cysticercosis infections, and those include the ELISA test and indirect hemagglutination. So this slide is showing you examples of a gravid proglottid for T. solium. And if you remember, we said that the lateral uterine branches, they can be about 15 to 30 for T. signata or the beef tapeworm. So if you look up through here, there's so many we probably can't even count them, but there's definitely 15 to 30 at least. When we look at the proglottid for T. solium or pork tapeworm, they won't have near as many, and you'll be able to see a huge difference between T. signata, our beef tapeworm, and our T. solium here in just a minute. This is a gravid proglottid, or one section of the beef tapeworm. This is an example of the scolex, or the head of our beef tapeworm. Here is our scolex with 4 suckers on it, so you can obviously see our 4 suckers. And our T. signata, or our beef tapeworm, is known to be unarmed, or the rostellum is known to be unarmed. So the rostellum would be protruding up here, and it would have hooks on it if it was armed, but the rostellum for signata is unarmed, so we don't see any hooks. And you'll be able to see a difference between the beef tapeworm and the pork tapeworm when we look at the pork tapeworm. But remember, T. signata is unarmed. The T. signata adult tapeworm looks very similar to the adult tapeworm that we looked at for our previous species. The head, or the scolex, would be over here. We're on the smaller end, and the tapeworm, each section would mature. These are probably immature sections, but as you go along the tapeworm, they're going to become more mature, and these are going to fall off, and these are going to have sexually mature organs in them, and they're also going to have fertilized eggs. Here is what our Tinea species of egg are going to look like. Again, we're not going to be able to tell the difference between our Tinea signata and our Tinea solium, our beef tapeworm and our pork tapeworm, but just looking at the eggs under the microscope, you would actually have to look at the worm itself, but there's hooks in the embryo or the egg, and those are these little lines that the arrows are pointing to. So remember, you're going to have this thick kind of shell, and you're going to have these hooks in your eggs for your Tinea species. Some additional notes for Tinea signata, or our beef tapeworm. The length of adult worms is usually 5 meters, but can reach up to 25 meters, and that's meters, so that's pretty long. Humans are the only known definitive host. Adults produce proglottids which mature, become gravid, detach from the tapeworm and migrate to the anus or are passed into the stool. And this is approximately 6 per day, so they can pass about 6 proglottids per day. T. signata adults usually have 1,000 to 2,000 proglottids. The eggs contained in the gravid proglottids are released after the proglottids or sections are passed in feces. T. signata can produce up to 100,000 eggs. So that's a lot of eggs. Now let's talk about Tinea solium. This is our pork tapeworm. The transmission is through ingestion of undercooked pork. T. solium is less frequently symptomatic than T. signata. The main symptoms are often due to the passage of the proglottids. Most important risk is development of cysticercosis. The life cycle is the same as T. signata. We looked at that life cycle a few slides back, and the pig was on there as well. So that's the main difference between signata and solium, is that the pig is the intermediate host instead of the cow. As far as lab ID, we've already mentioned this, but the eggs or the embryos are indistinguishable in feces between T. signata and T. solium. You have this thick outer shell, and you'll be able to see the hooklets within the eggs. Again, that thick outer shell. But again, you're going to be able to differentiate the species. As far as the gravid proglottid for T. solium or the pork tapeworm, you're only going to have about 7 to 12 lateral uterine branches. So not near as many as in T. signata. So these you might actually have a better chance at counting because you're only dealing with about 7 to 12 on each side. And this one is that pinkish color because there is a dye been used on the slide. And this one is, it doesn't present with that color because a dye has not been used. You should be able to differentiate species if you're able to see the scolex of T. solium versus the scolex of T. signata. The scolex of T. solium has 4 suckers as well, but it has what we refer to as an armed rostellum. So if you look over here, you see the 4 suckers. There's 1, 2, 3, 4. But here is the armed rostellum. You have these hook structures. And that's what makes it armed, or why we refer to it as armed. It almost looks like a crown of hooks, and this helps the worm hook to the intestinal wall of the host. Here's just another look at the scolex of T. solium. Again, you're going to have those 4 suckers, plus you're going to have the armed rostellum and all the hooks that are located on top of that rostellum that helps that tapeworm hold on to the intestinal lining. Some additional notes for T. solium. It is more prevalent in poorer communities where humans have closer contact with pigs or eat undercooked pork. T. solium is usually very rare in Muslim countries. Why? That's because they usually do not eat pork for religious reasons. But human cysticercosis is common in areas like Mexico and Central America. Adult worms can live for many years. Usually only one worm is present in the intestine at a time. Humans can serve as an accidental intermediate host if the eggs are ingested or released from the proglottid inside the GI tract. The larvae can migrate and lead to death if the racemose form develops in the brain or the cyst form from T. solium develops in the brain. This concludes part 1 of our cystode lecture. I will see you over in part 2.